Hello everyone. Welcome to another IBM webinar. I'm Morgan from IBM Technology and I'm glad you could join us today. In today's session, we'll be discussing intravital imaging of rat organs as cellular resolution in metabolic disease research. You will discover how intravital imaging in live rat models reveals real-time cellular and vascular dynamics across multiple organs, providing insights for metabolic disease research. As you follow Hayden's presentation, feel free to visit our website, ibmtech.com, and shop page ibmtech.shop to learn more about the imaging tools we'll be highlighting today. I'm pleased to introduce our presenter, Hayden, from the Technical Application team. With extensive hands-on experience in intravital in vivo imaging, Hayden brings deep expertise in this advanced technique at IBM. Please don't hesitate to submit your questions in the Q&A box at any time during the presentation. We will do our best to answer as many as possible during the Q&A session at the end. Hayden, if you're ready, please go ahead with your presentation.
Okay, thank you for the introduction. So before starting my presentation, let me share my computer screen first. Okay, so I think you could see my screen very well right now. So let me start my presentation. So, okay, thank you, Morgan, for my introduction again. So, hello, everyone. My name is Hayden, an application specialist at IBM Technology. So, first of all, I'd like to sincerely thank all of you for joining today's webinar. So, in this session, I'll begin with a brief introduction to IBM Technology's Intravital microscope, following by an overview of our newly launched system optimized for rat imaging, and then share a variety of application examples. In each section, we will also provide short virtual demonstrations ranging from simple abdominal organ imaging to window chamber implantation surgery, along with representative images of the results. Okay, so intravital microscopy is a high-resolution imaging technology that enables us to observe subtle biological changes inside a living animal down to the level of cells and tissues in real time. By comparison, traditional histology methods rely on fixed tissue sections. So while they provide detailed structural information, they only capture static snapshots, making it impossible to track dynamic processes as they unfold. So similarly, in vitro techniques using culture cells, it allows for controlled experiments, but fail to fully replicate the complexity of the in vivo environments, where cells constantly interact within their native tissue and vascular networks. Other in vivo imaging modalities such as PET, CT, or MRI are valuable for analyzing physiological changes at the tissue or organ level, but they lack the resolution needed to visualize cellular level events. But in contrast, intravital microscopy fills this gap. So it allows researchers to continuously monitor drug distribution, cellular interactions, immune responses, and disease progression directly within a living subject. So this unique combination of cellular resolution and real-time tracking provides a more accurate and physiologically relevant understanding of biological processes. So, as I mentioned previously, conventional techniques only yield raw resolution data since they capture images at the whole body or whole organ level. So, to overcome these limitations, we have developed a more advanced intravital microscope that enables researchers to track cellular dynamics, monitor drug distribution, and assess therapeutic effects, even in challenging organs such as the lung and heart in living animals. A defining strength of IBM's intravital imaging system lies in its clear departure from conventional in vivo approaches. Intravital methods typically require sacrificing separate animals at each time point, which inevitably introduces substantial inter-animal variability. In contrast, our system allows longitudinal imaging within a single living subject at precisely defined intervals, dramatically improving consistency and reliability and as well. So beyond static image acquisition, it enables real-time dynamic visualization of biological processes as they unfold, offering a far richer and more accurate perspective on complex physiology. To really understand how our body works, we need to see what's happening inside, right down to the level of individual cells. So this is especially true when we study the immune system, cancer, or how medicine move and act in the body. The intravital microscopy makes this possible. So it lets us watch these things unfold in real time inside a living animal. So our system in particular allows us to track how drugs travel through the body and how they affect cells life as it happens. So for example, on the left here, you could see the data from the sepsis model, where inflammation blocks blood vessels in the lung. And on the right, we show how our imaging system tracks oxygen, anti-cancer drug distribution, and transplanted cells moving through the body. So here you could see the range of intravital microscopes we have developed and optimized across different animal models. Some from a basic scale system designed for small animals like mice or hamsters to larger series that can even be applied to non-human primates. Our portfolio covers a wide spectrum without size limitations. So today I will focus on the IVM free space systems designed for small to medium animals such as adult rats, 3 weeks or older, ferrets, and guinea pigs. So along with this, I will also share several application results we obtained using this platform. The Ivan Technologies intravital microscopes are designed as all-in-one system, making them easier for users to operate. So these systems are simple to install and maintain, and they can be used without a darkroom, with the darkroom, allowing researchers to focus entirely on imaging and experiments without limitations from space or environment. So in particular, this free space model extends usability beyond small animals like mice to larger species such as ferrets and adult rats or rabbits, while maintaining the same all-in-one convenience. So at the same time, like our previous system, it still delivers outstanding intravital imaging performance through our in-house developed hardware and uh, software. These include the ultra-fast polygon scanner, real-time temperature monitoring system, motion compensation, and AI-based denoising software enabling high-resolution imaging of diverse organs in living animals. So among these features, the motion compensation system corrects for organ movement, so researchers can obtain sharp and accurate images even in high dynamic organs. And the real-time temperature monitoring system ensures that an animal's body temperature is continuously tracked and adjusted if needed throughout long imaging sessions. So this not only minimizes the physiological variability caused by temperature changes, but also maintains an ethically sound experimental environment. So finally, our soon-to-be released AI-based denoising software preserves true signals while effectively removing only the noise. So this provides cleaner, more reliable data that can be used directly for quantitative analysis. So later in this talk, I will also show you the example data sets processed with this software. So I would like to highlight some of the unique features of the new free space system compared to the previous confocal and multiphoton system. So most components, including the laser, lenses, and other internal option structures remain almost identical. So you could use both confocal and two-photon in the same this microscopes, not only to small animals, but to the mice or rat, ferret, guinea pig size animals as well. So in the earlier version, the stage measures about quite smaller, whereas in the free space system, the width has been nearly doubled. So this makes it possible to image the small or medium-sized animals imaging. And okay, so another feature is the open space design. So since the mounting stage is exposed, using the two-photon laser requires creating a dark environment with curtains or additional room modifications. But this open configuration also provides a major advantage, which it could offer much more space to attach external devices such as ultrasound equipment, treadmill, or some kind of behavior equipment if you need it. IVM Technologies intravital microscopes are designed not only for short-term studies but also for long-term imaging applications. So to make extended imaging more stable and reliable, we provide a variety of window chamber options. So by using these chambers, it's possible to perform PK/PD analysis as well, tracking disease progression or drug movement in real time within the same living subjects. So on this slide, you could see some of our most representative windows and chambers. So if you have a particular research interest in a specific organ, we can also develop and optimize customized versions of windows and chambers tailored to your study needs. So with our broad range of imaging systems that cover animals of different sizes, together with the adaptable windows and chambers, we were able to visualize a wide variety of organs in real time. So this makes it possible to apply intravital microscope across many research fields, from studying immune responses in vivo to following the progression uh, of cancer and other diseases, and tracking the drug delivery and mechanisms of action. So looking at some example datasets, you will see how our system could be used to monitor the immune responses triggered by different drug formulations, evaluate drug efficacy, visualize fibrosis, which is the therapeutic target of non-alcoholic fibrosis drugs, and image both the delivery and efficacy of various drug formulations in vivo. Okay, so right now I'm gonna describe some of the surgical procedures and followed by the imaging for visualizing different kind of immune responses or other cellular dynamics. Okay, so first, let me briefly introduce a simple surgical technique we use to image a rat kidney and then show you some of the imaging results we have obtained. Okay, first, we carefully remove all the abdominal hair of the rat using a clipper followed by depilatory cream. Then next, we make a small incision in the skin above the kidney to expose the organ and place a cover glass and directly on the surface after mounting the kidney carefully without harming or just disrupting it. And after that, we mount this red model in the free space system and then connect the cable for the stage's heating function and then gently move the tissue closer to the objective lens. So using the jog dial, we navigate until the kidney is positioned at an optimal location for imaging. Before the surgery, we prepared the rat model as illustrated on the left. So first, we injected fluorescently labeled red blood cells intravenously. In addition, we used our in vivo labeling kit to stain the vasculature in blue. So as you can see in the image on the right, this resulted in red-colored red blood cells flowing within the blue stained blood vessels. So because brightness can vary depending on tissue position and imaging depth, there is a difference between the first and second location shown here. So however, even at deeper layers, we were still able to observe the red blood cell flow and perform quantitative analysis to calculate flow velocity. So this means we can monitor changes in blood flow speed before and after inducing kidney-related inflammation disease or after administering therapeutic drugs to treat that kind of disease condition. And in this experiment, because the rat was kept alive, we were able to collect real-time data from multiple organs within the same animal model. So here, we exposed the inguinal lymph node of the same as the rat through a simple incision as well. You could see the exposed lymph node here. And this method allows us to directly visualize the neutrophil RBC, the red blood cell motility inside the lymph node in real time. And you could see also the green color immune cells here. So the fact that the animal remained alive made it possible to capture immune cell or some kind of blood cell dynamics with ease. So in particular, by labeling different immune cell populations such as T cells and B cells, we could also clearly distinguish their distinct motility patterns in the future. So next, we can move on to the second application. So moving beyond simple short-term imaging, we introduce a method for long-term imaging using a window chamber. So in this study, we implanted an abdominal imaging window into the abdominal wall and skin, which allowed us directly observe the liver over an extended period of time. So the procedure is carried out in a similar way as in mice and the imaging of the kidney in rats. So this rat was anesthetized with ketamine. So once anesthetized, the abdominal hair was completely removed using clippers and depilatory cream, and the skin was then incised to fit the size of the window. And next, the window was secured to the skin using a purse string suture technique, and the surgery was completed by implanting the abdominal imaging window and also attaching a rounded glass cover with adhesive. So after completing the abdominal imaging window implantation, this rat was placed on the free space system's mounting stage, and the abdominal imaging window was securely fixed with the holder. And the FITC dye was then injected via tail vein injection to visualize the blood flow, and also to maintain the physiological condition stable. A heating cable was also connected to the system for temperature control and monitoring. The stage position was roughly adjusted using the handles and then fine-tuned with a joke dial to locate the focal plane before proceeding with imaging. So similar to simple imaging methods, liver observation through the abdominal imaging window also provided stable imaging conditions. So this allowed us not only to capture deep images, but also to acquire deep and wide field images using imaging mosaic function. So with video level imaging technique, we could clearly visualize the blood flow, which also implies the possibility of measuring blood flow velocity. And now you're looking at the data before and after processing with the AI denoiser software. So within the free space system together with the denoiser software, I'm pleased to introduce these powerful tools that enable the observation and analysis of diverse biological phenomena in rats. So currently, we are expanding the pipeline to include the targeted cell labeling, imaging across different organs, and monitoring cellular responses in various disease models. So this pipeline will allow us to share an even wider range of data in the future, and also you are very welcome to ask uh, or help or need. Okay, so let's move on to the next study. So in here, we have induced septic diseases with administering LPS through intraperitoneal injection route, and we'd like to see the immune responses in lung and heart, especially the neutrophil. So first, we prepared two groups of SC rats, the control group and the sepsis model, induced by intraperitoneal injection of LPS. So to visualize the blood flow, we administered the FITC dextran, 2 megaton size one, and for labeling the neutrophils and blood vessels, we also administered the in vivo labeling kit based on the lectin and Lysx-G antibody system. So in this sepsis model, following the LPS injection, the acute lung injury occurred, leading to an increase in immune cell activity. So this is something we had already observed in mice, and here we were able to confirm the same phenomenon in rats as well, providing the idea that free space system would become the pivotal platform in intravital rat imaging in many research fields. So in the same breath, we additionally performed imaging of the heart. So although the data I will show here are not the videos, similar to the lung, we were able to observe both blood flow and distinct changes in immune cell behavior. Okay, so here is the last application session. The stroke or the microinfarction have recently emerged as important research topics in neuroscience, as they are closely linked to the cognitive decline and the onset of neurodegenerative diseases. So in particular, the activation of immune cells is increasingly recognized as a key factor influencing both the progression and recovery of these conditions. So however, there is still limited knowledge about how quickly these immune responses occur in vivo. So to address this, we set out to observe in real time the progressive recruitment of immune cells, especially neutrophils, in the brains of live rats after stroke induction. To implant the cranial imaging window on the rat, we first remove all the hair on the head as well using a clipper and cream. So after cleaning that area, the head is fixed onto the stereotactic surgical stage, and to protect the eyes, we apply ophthalmic ointment, and then disinfect the surgical site once more, just to be sure everything is clean and disinfected. And after that, the scalp is sent inside to expose the skull. And let me skip this part. Okay. So here we use using a drill tip of the desired size, typically 3 to 5 millimeter in diameter, we create an opening and remove the bone fragment. So a cover glass of the same size is then placed over the exposed cortex and secured in 3 steps using Loctite and the UV curable bonds, and finally the cement. So once prepared, this window remains stable from several days to over a year. So this durability makes it suitable not only for simple brain imaging, but also for long-term studies of neurodegenerative disease such as dementia or Parkinson's disease. So in this study, to model the hemorrhagic conditions such as stroke or microinfarction, we intravenously injected rose bengal and then applied a 561 nanometer laser wavelength to the targeted region. So this triggered the production of reactive oxygen species, which induced localized vascular injury. So as a result, thrombosis was formed, physically blocking blood flow in the vessel and thereby reproducing the disease condition. So first, to confirm that thrombosis was successfully induced, we injected FITC dextran to visualize blood flow. So within the white dotted box here, you could see that the unstained black red blood cells gradually accumulated over time. So although the data is not attached here, in the actual real-time video recording, we could also observe a progressive slowing of blood flow, which provided an early indication that the disease reduction had been successful. So after confirming that stroke induction using Rose Bengal could also be achieved in rats, we next sought to observe the immune cell behavior, the neutrophils, by applying our Lysx3-based in vivo labeling. So initially, no neutrophils were present in the cortex region. However, beginning around the 40-minute time point, we could observe them gradually migrating toward the cortex, as indicated within the white dotted box here. So as I mentioned earlier, this supports the understanding that immune cells act dynamically in conditions such as dementia, as importantly that they begin to act at a relatively early stage of disease progression, not only the dementia, but also the microinfarction or stroke. Okay, so let's wrap up my presentation now. So this is my last slide here. So with IVM technology, you could not only visualize the hair, kidney, liver, but also almost all organs from the rat, not only the mice. So we are now able to meet the needs of researchers not only in mouse, but also in rats from almost all organs, including cancer or immune cell reactions from various disease models, and also some kind of drug efficacy or biodistributions in living body. Okay, so thank you for your attention, and please, if you have any questions, you could interrupt me from now.
Okay, thank you, Hayden, for the great presentation. Now let's begin the Q&A session. So our first question is, hello, thank you for the webinar. Our lab use both mice and rats. Do we need to purchase two separate systems to perform intravital imaging on both animals?
Okay, so thank you for the first question. So we have also optimized and put that model, which is called CM3 or CMS3. So you could utilize confocal and two-photon laser both in the same system for visualizing the mice. But also you could use that system under 3-week-old mice because the body size is quite small. But if the rat gets older and older, the size gets bigger and bigger as well. So in this case, you definitely and rightfully need to use the free space system. But for just in case, we also have another adaptable uh, stage for the mouse image. So if your lab studies both rats or mice, you could utilize the free space system. So after rat imaging is done, for the mouse imaging in the next step, you could just simply put another some higher step like a plate, and then you could finally free to use to image the similar cellular activities from the mouse. So my answer is you could use this free space system for both mice and rats.
Okay, thank you. And let's move on to next question. So second is, how do you perform in vivo staining for rat surgery?
Okay, so come again, please. I'm sorry.
How do you perform in vivo staining for rat
Mhm.
surgery?
Ah, okay. So first, our system is based on fluorescent signal detection. So you could utilize your own transgenic mice, rats, expressing fluorescent signals of, for example, GFP, RFP, or Cy5, including the CFP or second harmonic. But in other case, we definitely label the target cells or molecules with the fluorescence. So we also provide the in vivo labeling kits based on various antibodies. For example, CD31 for endothelial cell imaging or Ly6G for neutrophil. Rightly, you could because you did see the result images from my presentation. But in other case, if you have some special target cells in your study, you could also purchase the DIY kit. It supplies the various reagents and the fluorescence, which is quite confirmed that it's quite compatible with our system. So in that case, you could choose just the wavelength, for example, the red fluorescence or blue fluorescence. Then you could conjugate the antibody of your own antibody and the fluorescence. So in that case, you could easily label your target cells in your lab with just only a few hours, I think. And for the surgery, definitely we need to slightly open the skin with a simple incision for the terminal or short-term imaging. But in other case, for the long-term imaging, as you could see in the previous slide, you need to do some kind of uh, surgeries for implanting various windows. So first, uh, okay. So one of the attendee asked the method for anesthetizing the rats. So first, in our lab, we mostly utilize the ketamine to anesthetize the mouse. But in other case, most of the researchers, especially in the States or the European countries, they are willing to utilize isoflurane gas. So we have the free space system is quite open uh, space system. So you are very welcome to adapt many tubings or adaptables to anesthetize, visualize, or induce the diseases. So as well, we could also put the anesthesia mask on the face to uh, make the rat go to sleep. So you could utilize both ketamine, the injectables, or ISO as well. And also, if you administer the labeling kit, it the first and signal stays over several hours. For example, maybe from 6 hours to 24 hours. So for the one-time imaging, just one administration of the labeling kit is enough. But if you are trying to image uh, several times repetitively, like with like a several days or a month interval, maybe you could inject again before the imaging. And also, yeah, one of the attendees asked a very important question. So the imaging depth. So typically, with confocal laser, we could go deep into like around 200 microns. But in terms of the red color organ imaging, the spleen or liver example, they have many cells inside of the organ. So in that case, maybe 150 to 200 microns. But with the two photons, we could go deeper, like a 500 microns. But for example, the brain is quite white, not the red. So in that case, we could go around 700 to 1000 microns. And our system is the closed and also not the free space system. the closed system, like a CMS3 or CM3, the previous models, they have also two holes for isoflurane inlet or outlet. So even if in the closed system, you could also utilize the gas anesthesia system as well. Okay, so they kept the questions are they keep popped up, but okay, like I said before, to be respectful of your time, I'm gonna get back to you as soon as possible after this webinar is over. Okay, thank you for the questions, and I hope to get back to you soon.
Okay, thank you everyone for your questions. The webinar recording will soon be available on our YouTube channel. If you have any more questions or if we couldn't address your inquiry during the Q&A session, please feel free to reach out to us at information@ibimtech.com. And as I mentioned earlier, we have recently launched the iBeam shop, where you can find imaging accessories, bronze dyes, and training programs. By registering an account, you can access certain benefits applicable to your purchases. Thanks again for joining us today.